Light waves exhibit interference. When two waves meet, their amplitudes add together. Constructive interference occurs when waves align, while destructive interference cancels them out. Diffraction occurs when light waves encounter a small opening or object. Instead of traveling in a perfectly straight line, the wavefronts bend and spread, creating a characteristic pattern. This bending creates the diffraction limit. Because light spreads out, a point source of light is seen as a central spot surrounded by rings, known as an airy disk. This sets the fundamental physical barrier to our resolution. Refraction is the phenomenon where light changes direction as it travels between materials of different optical densities. The refractive index of the material determines how much the light ray bends at the interface. By shaping glass into lenses, we harness refraction to focus divergent light rays into a single image point. A lens uses its curved surface to precisely manipulate light. By leveraging the principle of refraction, it bends parallel light rays toward a specific focal point. By controlling the degree of refraction, lenses focus light to a point, allowing us to form a magnified image of tiny specimens. This process is the foundation of microscopy. It transforms the light scattered by a specimen into a coherent, enlarged visual representation that our eyes can resolve. The electromagnetic spectrum spans from high-energy gamma rays to long-wavelength radio waves. Visible light occupies only a tiny fraction of this vast range. In microscopy, we primarily use the visible light slice between 380 and 750 nanometers. Shorter wavelengths on the left provide higher resolution, while longer wavelengths on the right offer less detail. By manipulating these frequencies, we can reveal structures invisible to the naked eye. A prism demonstrates dispersion, where white light is separated into its constituent colors. As light enters the prism, each frequency refracts at a different angle because of its unique interaction with the material. Because blue light bends more than red, the white beam disperses into a visible spectrum. This ability to manipulate light is fundamental for specialized microscopic imaging. Understanding dispersion allows us to filter light for high-precision imaging systems. Fluorescence and phosphorescence are photoluminescence processes. Atoms or molecules absorb a high-energy photon, jumping from a ground state to an excited state. In fluorescence, the electron quickly returns to the ground state, emitting a photon of lower energy. This happens almost instantaneously. Phosphorescence involves an intersystem crossing, where the electron gets trapped in a triplet state, causing a delayed emission of light over a longer period. In modern microscopy, this is transformative. We use fluorescent labels to tag specific proteins, allowing us to visualize microscopic structures with high specificity. Magnification is the process of enlarging the apparent size of an object. It allows us to see features that are otherwise too small for the human eye. However, magnification alone is insufficient. Resolution is the true limiting factor. It is the ability to distinguish two closely spaced points as separate entities. If you magnify an image without sufficient resolution, you get what is called empty magnification. The image gets larger, but no new detail is revealed. The image remains blurry. A superior microscope prioritizes high resolution. By increasing the numerical aperture and using shorter wavelengths, we achieve the clarity needed to unlock cellular secrets. Resolution is defined by our ability to distinguish two distinct points as separate entities. When points are too close, they blur into one. One way to improve resolution is to shorten the wavelength of the light source. Shorter wavelengths interact more precisely with small structures. The second factor is the numerical aperture. Increasing the angle of the light cone entering the lens captures more detail, drastically improving clarity. By optimizing these two variables, we bypass diffraction limits, allowing us to see the fundamental details of microscopic samples. In the late 17th century, Antonie van Leeuwenhoek transformed our understanding of the microscopic world. Unlike his contemporaries who used compound lenses, he mastered the art of grinding tiny, near-perfect glass spheres. Using these lenses, he became the first human to ever witness bacteria and protists. He called these moving organisms animalcules, documenting a hidden universe teeming with life in a single drop of pond water. His work bridged the gap between the unseen and the observable, proving that there is a vast complexity beneath our sight, forever altering the path of biological discovery. In 1665, Robert Hooke revolutionized biology by using an early compound microscope to examine the structure of cork. Under the lens, the cork appeared as a dense network of tiny rectangular partitions. Hooke named these structures cells, as they reminded him of the small, partitioned rooms, or cella, in a monastery. This discovery laid the foundation for the cell theory, identifying the fundamental building blocks of all living organisms. A simple microscope utilizes a single convex lens to magnify an object, functioning much like a high-powered magnifying glass. The light rays pass through one lens, creating a virtual enlarged image. In contrast, a compound microscope employs a two-stage system. The objective lens gathers light to form an intermediate magnified image, which is then further magnified by the ocular lens or eyepiece. By combining multiple lenses, compound microscopes effectively multiply magnification powers, allowing us to resolve fine structural details that a single lens simply cannot capture. Ultimately, while simple microscopes are perfect for basic observation, compound systems are the cornerstone of modern cellular biology research. Confocal microscopy improves upon traditional imaging by using a focused laser to scan a specimen layer by layer. By placing a pinhole aperture in front of the detector, the microscope rejects light from outside the focal plane, significantly increasing image contrast. This technique provides true optical sectioning. By capturing thin horizontal slices at different depths, known as a Z-stack, advanced software can reconstruct highly detailed 3D structures from thick biological specimens. Two-photon microscopy uses longer wavelength infrared lasers. Excitation happens only when two photons strike the same fluorophore simultaneously. Because this only occurs at the tight focal point, it enables deeper imaging into tissue with minimal damage to live samples. In traditional light microscopy, we are limited by the physical wavelength of visible light. Light waves are relatively long, which restricts the level of detail we can capture. Electron microscopes replace visible light with a high-energy beam of electrons. According to quantum physics, these electrons exhibit wave-like properties with extremely short wavelengths. Because the wavelength of an electron beam is thousands of times shorter than that of visible light, it allows us to overcome the diffraction limit, achieving resolution far beyond what light can reveal. In practice, this instrument requires a vacuum environment to prevent electrons from scattering against air molecules. A high-voltage source emits the beam, which is then focused using precise electromagnetic lenses. This technology enables researchers to observe structures at the nanometer scale, providing unprecedented insight into the building blocks of biological and material systems. The transmission electron microscope, or TEM, functions by passing a beam of electrons directly through an extremely thin specimen. This allows us to map the internal ultrastructure of biological samples or materials. In contrast, the scanning electron microscope, or SEM, uses a focused electron beam that scans back and forth over the surface of a specimen. The resulting interactions provide high-detail, three-dimensional topographical views. While TEM excels at revealing internal components by looking through the sample, SEM is the tool of choice for observing surface structures and 3D morphology. Scanning probe microscopy represents a departure from traditional optics. Instead of using light to image a sample, it uses a physical probe to feel the surface of a specimen. In atomic force microscopy, a sharp tip at the end of a flexible cantilever scans the surface. As it moves, the probe detects the subtle forces between individual atoms. A laser reflects off the back of the cantilever onto a detector. This allows the microscope to track even the tiniest vertical deflections as the tip encounters atomic features. By mapping these mechanical or electrical interactions, researchers can create precise 3D topographical maps, allowing us to visualize matter at the scale of single atoms. Before a specimen can be viewed under a microscope, it must undergo preparation to maintain its structural integrity and increase visibility. First, fixation is used to stabilize cellular structures, effectively stopping biological processes and preventing decay. Next, mechanical sectioning uses a microtome to slice the sample into extremely thin layers, allowing light or electrons to pass through. Finally, staining adds contrast to transparent tissues, highlighting specific organelles or chemical features. These three steps, fixation, sectioning, and staining, are the cornerstones of preparing reliable and informative microscopic samples. Electron microscopy requires specialized preparation. For transmission electron microscopy, or TEM, the challenge is that electrons must penetrate the entire sample. To achieve this, biological specimens are embedded in hard plastic resin and sliced into extremely thin sections, often less than 100 nanometers thick, using a diamond blade on an instrument called an ultramicrotome. For scanning electron microscopy, or SEM, the process is quite different. SEM images rely on electrons reflecting off the surface of the specimen. Because biological samples are poor conductors, they accumulate a surface charge that distorts the image. To fix this, samples are sputter-coated with a thin layer of conductive metal, such as gold or platinum, ensuring a clear, high-contrast image. Proper preparation is the bridge between a raw biological sample and a high-resolution scientific image. Fluorescence microscopy relies on tagging specific structures with fluorochromes. These molecules absorb light at a specific wavelength and emit a lower energy glow, allowing us to visualize structures with incredible precision. We start by targeting specific cellular proteins. We apply fluorescent probes that bind uniquely to these structures, acting like biological beacons. When we illuminate the sample with excitation light, the fluorochromes absorb the energy. This causes them to reach an excited state. Almost instantly, they release this energy as emission light. This is the characteristic glow we observe, filtering out the background to highlight only the tagged structures. The result is a high-contrast image where only the structures of interest remain visible, enabling advanced cellular analysis. Modern microscopy has entered a new era, driven by artificial intelligence. By integrating deep learning, researchers can now reconstruct high-resolution images from noisy or low-light data. This integration solves a critical problem: phototoxicity. Intense light can damage living cells. AI allows us to capture high-quality images using significantly lower light exposure, preserving specimen viability. Furthermore, AI algorithms now automate the identification and classification of cells, enabling researchers to process massive datasets in seconds, which once took weeks of manual work. The future of microscopy is shifting from static observation to real-time dynamic mapping and multimodal integration. Correlative light and electron microscopy, or CLEM, combines the specific labeling capabilities of light microscopy with the high-resolution structural details of electron imaging. We are moving beyond snapshots of dead cells. Real-time dynamic mapping allows us to observe molecular interactions as they occur within living, complex systems. The next decade will see lab-on-a-chip portability and AI-driven image analysis, making powerful diagnostics accessible everywhere.